I don't have an outline slides. I'm going to just tell you that I am going to give you a little bit of history at the beginning of how I got to involve in this line of research, and then my talk is kind of broken down, roughly speaking, in two set of studies. One that focused more on the neural correlates of language in infants at a high likelihood for developing autism, and then another series of studies still in this population, but studies that I think are informing our understanding of the mechanism that may give rise to the autism symptomatology very early on in life. So I'm going to start by actually acknowledging the person to whom I owe a lot in terms of steering my career down this path, the autism path, and that is Marion Sigmund, who I know many of you know here, knows very well. I was not one of her trainees, but she remembered as I started my postdoc that it had something to do with some new technology. You can study the brain, and we ran into one another in front of our Neuropsychiatric Institute, and she says NIH is going to fund some centers of collaborative a program for excellence in autism research, and one project needs to be on the neurobiological basis. So can we get together and talk with your mentor, and the rest is history. And my mentor was Susan Buchheimer. She's been the pillar of our community, and I think the pillar also of the major pillar in the autism community. It's interesting that in our third program grant, in the third iteration, she brought me back into the infants because we obviously needed to have some common lines across a different project, and we talked about adding some behavioral measure in the infants that I was using in my MRI, and then when her health began to decline, I got more sucked into this work, and when it was time for us to submit the next project, when she had stepped down as the center director, we really felt like we didn't torn between and having a baby project or not, because she was no longer with us, but at the same time we wanted her legacy to continue. And as luck would have it, I was asked to review a paper that it's this paper that showed that you can actually study the neurocorrelate of language in neonates 2 days old in the study before they even were home from the hospital. And that got me really excited because you mentioned, she mentioned my language was one of the main interests early in my career, and here you see an example of the beautiful activation maps that you can get in babies that are just 2 days old. And you could also, in the other, here you can also see that you can already trace some of the tract. And that's perhaps not too surprising because babies 7 months in utero already process and respond to language. So at that point, I got excited and I said, okay, well, maybe we'll go down that path, but let me try and collect some pilot data. And lo and behold, I got some beautiful maps. I don't know how many of you here are neuroimaging folks, but this is one single baby to 8 weeks old, and these are resting state functional maps that inspire me to go down that path. And as they say, the rest is history. I got convinced that we can not only look at the neurocorrelates of language, but you can also look at the network level at a variety of networks, not just the sensorimotor, auditory, and visual network, which we know develop early on, but even some higher level networks like the default mode network, although it's not fully resembling the adult form, with also the anterior cingulate until later on, and the salience network, to which I'll return. So I'd partner up in this effort with Shafali Jeste, who had just recently joined the faculty at UCLA, and with Scott Johnson, who is a developmental psychologist who has done a lot of work in using eye tracking methods. And so we went in with a project that had really focused on the first year of life, because I don't need to tell you guys, a lot of the work has been done here by the best example of the baby siblings work has been done here. The behavioral markers that have been identified typically emerge after in the second year of life, 12 months and on. At least at that point, that's where the field was. And so we thought maybe the brain can tell us something that it's harder to detect in the first year of life by looking at what babies can do. After all, they still have somewhat of a limited repertoire. So we did MRIs, and it involved both stimulus evoked. I don't use the word task because babies are asleep. We did resting state, diffusion tensor imaging, and Shafali led the work on EEG. Scott had some studies on eye tracking, a host of behavioral and clinical assessment at different time points in the second year, and also some of it earlier. And also we did some genotyping, although we still have to try and relate any of the genotype information. So this is not an audience where I need to describe what the infant sibling approach, but I put these slides in for one reason, and actually, well, maybe two. So the national prevalence continues to increase, but the siblings that are born in a family where there is already a child have a much, much higher recurrence, and even if the prevalence is now almost like 2% or a little bit more, the recurrence in this population is much higher. And what the point is also that in addition to the babies who go on and get a diagnosis, there is quite a larger group within this population who end up having suboptimal outcome and having either developmental delays, language issues, and whatnot. But the goal of this approach was to ultimately compare children who do get a diagnosis to the children who do not, right? And I'm sorry to tell you that after 10 years of being in this business, we still do not have a large enough sample where we can actually break down our high likelihood group into two distinct groups. So all of the work that I'm going to show you today, it's really looking at risk by comparing the high likelihood, the babies who are at high likelihood versus the one who do not have that. And at the best, I'll show you some dots that once we knew who were the ones that got diagnosis. So after this maybe too long introduction, the first part of my talk, as I said, will focus on early language. I'll be showing you a study on the maturation of the dorsal language tracts in 6-week-old infants. Then I'll talk a little bit about functional connectivity of language networks as they change from 1.5 to 9 months of age, and also some work that I really love that tap into not just the neural correlate of language processing, but also tapping into implicit learning. And lastly, a study that it's more recent, and I was debating whether it fell more in the realm of the tapping into mechanism or being language. I ended up putting it here in part because it still ultimately deal with the communicative language trajectories, but okay, without further ado, let me just jump in. And the first study was done, actually the first 3 studies that were done by a fantastic graduate student of mine that is now a project scientist working for the HBCD study, which is the large-scale study funded by NIH that will enroll over 7,000 babies before they're born and follow them for 10 years. And so I showed you earlier that that study showed that you can actually see language tract already early, very, very early in development. We know that, and we also know that a little bit later in development, infants who then receive a diagnosis, as well toddlers with autism, show altered structural connectivity of these tracts. So the question was, can we actually detect differences in this group? And we focused on 2 tracts here. You can see one that connects the arcuate fasciculus that connect the posterior superior temporal gyrus to premotor cortex, and then the superior longitudinal fasciculus that actually connect the same region all the way to Broca's areas. And the arcuate fasciculus is one of the tract that it's very important for sensorimotor integration of language function, and typically the superior longitudinal fasciculus is involved in some of the higher level language. So very quickly, just this is we collected the data at 1.5. We had language measures, and here we focused on the MacArthur Communicative Development Inventories at 18 months, and then we had for some of these babies, ADOS is done at 36 months, a few we missed. I'm not going to bother you with some of the pre-processing step, but basically we focus on one particular measure that you'll see abbreviated as FA, which is an index of integrity of white matter tracts. We know this is increasing with age, so higher number is good. And we also know that in other studies it's been found to be lower in, again, individuals with autism. And we also looked at a laterality index because prior studies have also seen in toddlers with autism some atypical pattern of lateralization. Typically, you see greater activation in language region in the left hemisphere, and also, in terms of language tract. So this is how we traced it. I mean, we put all of the babies in a standard space. I hope you can. They look very similar, but if you pay attention to what is happening here, you can see that this involves definitely more of the precentral regions, and the tracts extend more anterior for the superior fasciculus. And now going into the results, you can hopefully see here that when we're talking about the anterior fasciculus, there were no significant differences. But in the superior longitudinal fasciculus, we did actually see that the typical likelihood infants show more robust superior left fasciculus FA. What we did then, we tried to find out whether this actually relates to any measure of behavioral measure that we had, and lo and behold, we did find that the more robust the left tract at 1.5 of age was actually predicting, or the receptive language advantage measure that we derived from the MCDI. So I don't know how many of you here will know what I'm talking about. So very briefly, babies typically comprehend many more words early on than they can produce, and a previous study had shown that this receptive advantage is actually decreased in babies who later develop autism. So that's why we picked this measure. Then I mentioned that we also wanted to look at laterality patterns, so we had computed the lateralization index, and what you see here is that for the anterior fasciculus, the difference was not significant. There was a trend, but there was a significant difference for the superior longitudinal fasciculus, and also if they combined, there was a significant difference, whereby the low likelihood babies, the babies at low likelihood of developing autism had a laterality, they had a less right lateralized language profile. Now, I just meant, we all know that language is a left hemisphere function for most of us, but actually early in development, you actually see a lot of right hemisphere involvement. So we take that this is the beginning of a shift, and then the babies that had an elevated risk for developing autism were a little bit further behind. So we then looked at whether this predicted or was related in any way to diagnosis, and indeed we did find that this more rightward lateralization, being so further down here, did actually predict ASD symptomatology in this group at 36 months of age. Okay, so what does this mean? And well, when neuroimaging, you never know what comes first, the chicken or the egg, but we hope that this may, we don't hope, but we think that this may have something to do in terms of hindering some of the development that we see, or the delay in language that we see later on. But I would love to be able to replicate in a much larger sample. This was our first study, and so we have not yet benefited from the second wave of babies. Janelle also focused then on functional connectivity. So when we're talking about DTI and tracts, we're talking about really the structural tracts that connects different language regions. Functional connectivity is just looking at networks of regions that tend to activate at the same time, subserving a similar task. So they need not to be structurally connected, but nevertheless, they're they tend to work together and to subserve, a variety of functions. Here we had resting state collected both at 1.5 and at 9 months of age. We used two different approaches. We first did a seed-based connectivity approach, a bottom-up, where we basically started out with our seed in the eschal gyrus, which is the primary auditory cortex. After all, language is processed in that modality, and we just looked at where activity vary in tandem with activation in these regions. And then we also used a slightly different network connectivity approach to compare the groups, which obviously you may infer when you're looking at a 6-week-old baby, the brain is much smaller than the one in babies that are 9 months old. And so we have tried to avoid having that issue of having to choose which space to put them in to actually look at brain differences. So just glance at this picture, you notice a lot of pink, which means that both groups had a similar connectivity pattern. You also see a little bit of blue and some red there, and while they look fairly similar, it's important to notice, to be reminded that, again, a lot of the high likelihood babies will nevertheless have a typical development. So there were actually some differences despite those similarities, and what you see here is that the connectivity with the left eschal gyrus was higher in the 1.5 month old babies in the typical likelihood than in the high likelihood. So sensorimotor function are very important in language. It's ultimately something that is good for the language region to be connected with motor regions. And in 9 months, we actually see a different pattern. Now here you have the high likelihood babies who actually show greater connectivity between the auditory cortex and other primary sensory region, in this case the somatosensory cortex. And that is actually one of the things that we see in several other studies in adults with older kids with autism, where there is hyperconnectivity within primary sensory cortices, kind of like a reverberation within that system. And then at 9 months of age, there was also a difference in the between the group comparison, where the typical likelihood babies had greater connectivity between, again, primary auditory region and other language regions. Then when we did look at comparing the two groups at each individual times, I'm not going to really go into explaining every single square that is going to be highlighted, but if you don't see a star, it means that there were no significant differences. At 1.5 months of age, the pattern of connectivity between all of the language-related regions we had chosen, as well as the thalamus, because that is a major sensory gating, did not differ significantly. The square looked like there was a difference, but it didn't survive comparison for the many correction for the many comparisons we ran. At 9 months, there were a few differences, and they can be roughly summarized as there being less connectivity with thalamic regions and increased connectivity between frontal and temporal regions. Then we focused on changes within each group between 1.5 and 9 months of age. And you can see that in the high likelihood, high-risk group, there were some changes. All of those asterisks in the notes, changes where there were significant changes, but nothing in comparison to what we see in the low likelihood or typical likelihood group that has, I hope, a striking difference in terms of there being many more changes. And by the way, I don't have the slides, but there was also significant interaction in many of these regions, suggesting that there is already something quite different that is happening over that time period where the high likelihood babies fail to establish stronger connection within language networks. The third babies in the dissertation of my student was a study that was kind of dear to my heart, because that's what brought me back into the infant literature. And I don't know how many of you are developmental psychologists, but there was a very pioneering study at that time that showed that infants at 6 months of age can parse a continuous stream of speech by using just statistical regularities in the input. Obviously, don't tell babies what to do, right? You're just observing their behavior. And we all have this biased perception where we think that there are pauses in between every word that we uttered. But in reality, as you can see from this spectrogram, there really are as long pauses within words than there are between words. We perceive those pauses because we already know what the words are. But when you're a baby and you know nothing, you actually cannot know, and you cannot attach any meaning to any word unless you have identified a particular, combination of syllable as being a word. And if you ever experience the impression that a foreign language to you, that people speak very fast, it's just because you cannot detect those boundaries. So anyway, as I said, this was something that was done originally in behavioral studies with babies, showing that this is something that is happening even earlier now. We know that in 6-8 months of age. And it's just basically saying that it's looking at the probability of syllable reoccurring, and as you can hopefully deduce from that little burp there is a much higher likelihood that certain syllables will follow one another when they're part of a unit that conveys meaning, when it's a word, than in between. And further studies, again, in behavioral studies, show that this is actually one of the many cues that babies can use, and the other one that comes online around 9 months of age is actually to focus on which syllable is being stressed. And I think in English, 80% of words have the stress in the initial syllables, and by now babies have begun to realize that it's a potent cues. So this is a very long and complicated paradigm. I'm not going to play any of these, but just suffice to say we created a paradigm that used continuous stream of speech where all of the syllables were matched and there were different set of syllables used so that the babies would never hear the same one. But in what we call the stress language condition, there were both statistical regularities and stress cues that can guide the parsing of that speech. In the unstressed language, there were only statistical regularities, and in the random syllables, we made it as random as we could so that it would be impossible. Now, when you look at those words like that in different colors, you go like, oh yeah, well, what's difficult? If I had shown you those that slide and then played them, you could hear the word because you've already seen them. But if you don't know what they are, you can be listening at it for 20, 40 minutes and not be able to tell them apart at a conscious level. But your brain and the baby's brain can actually do that work. We had validated this paradigm in both normal adults and in a bunch of kids. And then we also did a study in neuroimaging studies in individuals with autism, in youth with autism, and found significant difference there. So the beauty about this paradigm is that it allows us to, like any other language task that people had used before, to compare activity during the exposure and versus the silence baseline condition. But it also allows us to look at the neural signature, if you will, of this implicit learning that it's happening, of this computation of transitional probabilities in the identification of this of the words, because we can look at how the signal changes over the course of listening to each of these languages. So because everything is counterbalanced and everything's the same and they just keep repeating the same syllables, the only thing that is actually changing as you're hearing those 2.5 minutes of gibberish is your brain processing those cues, okay? This was done in 9 months because I mentioned that that's one of the things, that's the time when one of the cues becomes more readily deployed to process this task. We had language measures. This is just a map showing activation across the different conditions for the two groups, and they looked relatively puny, and that's the problem for the neuroimaging folks among us when you have to threshold now quite stringently, and I swear to God they were much more, they were larger and encompassing even more of the whole language network. So if you were just looking at a 2.3 Z of 2.3. So there were also a few differences, and I know they're so tiny that it's hard to see them, but the low likelihood babies actually did show greater activity in this left temporal regions, and there was also a difference between the two groups, and only for the stress condition, the one that contained those speech cues, that babies that have started acquiring language have realized already are good indices of when words begin. But more importantly, we were interested in looking at these changes over the course of the presentation of these speech languages. And here we did see that the typical likelihood babies did show signal increases over the course of listening to the stressed language that were greater than the signal increases that you would see during the course of listening to the unstressed language. And by the way, this left hemisphere cluster here is very reminiscent of what we have seen in adults and 6 years old all the way up to teenagers. And we also did see that if one extracts the activation in this area, it was actually predictive of better language outcomes, at 3 years of age. Now, we also compared the two groups, and we did find that very similar regions, same in the left temporal lobe, did also show that the typically likelihood babies had greater increases than the high likelihood babies for this, comparing the condition with the speech cues, the social cues, if you will, versus just the bare bone statistical regularities. And when we extract a parameter estimate of activity from this cluster, which is also where we have found differences in our study with individuals with autism, we actually saw that it was predictive of both the MacArthur expressive score at 36 months of age, and also the ADOS severity score. So basically, what you see is that even if this is a region where overall the group at high likelihood for developing autism had less signal increases, the variability in their responses was such that the babies that had the higher signal, the more normative patterns, were the ones that had better expressive scores, and also vice versa, the ones that had the least amount of signal increases in this area were the ones that were doing more poorly in terms of autism symptomatology. I didn't say it, I should have said it, that as I was going through this differences we see particularly related to this stress condition where you have not just the statistical regulators but also the speech cues, does kind of go along with the notion that speech cues are less salient to the babies who then go on to have to develop autism. So I mentioned that this other study on the cerebellum is actually a study that I debated whether it was a study on mechanism or a study that I should present to you in this context. But I figure it belongs here. And for those of you who think about the cerebellum as just the motor function, I think in the past 20 years or so, we have come to appreciate that it actually does much more than that. And in particular, there is one region, the right crus one, that is being shown to support both language function and also social processes like mentalizing. And this is also a region that prior work have found atypical connectivity in older youth with an autism diagnosis. So one of my the best, if I had to say, but I shouldn't draw a comparison, one of my brilliant research assistant took the lead on this study, and she's now doing an MD, PhD at Harvard and the Harvard-MIT combo, and she really decided to take a slightly different approach, and this was like a testament to her creativity. Instead of comparing just our high likelihood and low likelihood groups, she decided to use a different approach, cognizant of the fact that, as we mentioned earlier, there are really at least two groups in which you can split the high likelihood groups in a group that would develop autism and a group that may not, right? And so based on some of the prior work that showed that you can actually use some bottom-up clustering to separate, to tease some of the heterogeneity, she followed the same approach, and we ended up splitting our larger group of both typically low likelihood and high likelihood into 3 different cohorts. And this picture looks slightly different than the one before, in part because that one was based on T-score, so it was showing the decline, and this one is based on row scores, and so it's actually showing a pattern of increases. But I just want to draw your attention to the fact that the 3 groups did not really look any different at 6 months or at 9 months or at 12 months, and indeed all the way up to 18 months, the differentiation between what we ended up calling the late blooming group that then caught up with the typical group by 36 months was really not different from the group that would show a delayed trajectory across the board. Okay, and we validated this, this grouping, if you will, with a number of analyses, but here you're just looking at the proportion of babies in each risk group by the cohorts that we identified, and it's not surprising that the majority of the high likelihood babies were assigned to the delay group, and there were also many more males in this group, and their language, not just their language score, but their visual reception scores on the molars were also different. And parallel to that, in the typical group, there were many more, obviously, of the babies with typical likelihood for developing autism. So then what we did, that grouping was based, I think I didn't mention it, it was based on the receptive language scores on the Mullen, but we had many more measures, and so we tried to validate this. And we've indeed showed significant group differences in terms of vocabulary development from a different instrument. This is parental reports on the CDI, so the groups differ in terms of word comprehended, and also in terms of words produced, but not until an 18 month of age. Then we also looked at differences in joint attention, which was assessed with the SAS, and both at 12 months of age as well as at 18 months of age, there were significant differences between all of the groups, which made us feel good. And last but not least, we also find significant difference in autism symptomatology. So the groups now differ based on the ADOS-T done at 18 months, particularly the social affect subscales, and also at 36 months. Again, you saw that there were now differences between the delayed and the other 2 groups, but the difference between the late blooming and the typical group had now disappeared. So by 36 months, the late blooming group had fully caught up. The analytic approach for our imaging data here was a little bit different. Like we did earlier on, we put all of the babies into our analysis because the number of babies in each of those 3 categories was not that large to do a group comparison from kind of like using a bottom-up approach. We use the RICRUS-1 as a seed to look at basically where connectivity across the entire brain. And once we have done that, we applied a region of interest approach where we basically only extracted parameter estimate or indices of the strength of the connectivity between the cerebellar region and a variety of cortical region that and subcortical that we had chosen based on prior evidence of involvement in both language processing, social mentalizing, and also evidence of differences in strength of connectivity between individuals with ASD and typically developing individual in prior studies. So at this point, we basically, I hope I made it kind of clear. So we start with the cerebellum. We're looking at connectivity across the board, but then across the entire group, the larger group of infants. Then we extract the parameter estimate of connectivity from this regions that we knew had been implicated before in the processes that we are interested, or that show differences between individuals on the spectrum and those who are not. And now we finally can do the comparison between our 3 cohorts. And lo and behold, what you see here is that we found significant differences in both connectivity between the right CRIS1 to prefrontal cortex and to the SMA. And what you see, this is the pattern that you'll see also in the next slides, is that it was the group, the delayed group, that differed from the other 2, but the other 2 groups, the late blooming and the typical group cohort, did not show any difference. The same pattern, as I mentioned, held also when we looked at connectivity between the right CRIS1 and the thalamus, and also right CRIS1 to the basal ganglia. And so now you can say, okay, why should we matter? Well, we should matter because these are data collected at 9 months of age. The imaging data are collected at 9 months of age, and these groups are not different at the behavioral level until, well, they could have been earlier, but definitely by 18 months of age. As you can see here, the late blooming group was still not showing any difference. So you have something that predicts whether is able to differentiate the kids for whom you really have to worry about versus the one that you don't need to, because they're going to eventually catch up. Okay, why did I say I debated whether to talk about this here or to talk about mechanism? I don't think I'm going to have the time to go over the details, but the cerebellum is actually a very important region at the level of thinking about its role in terms of mechanism, because it's really involved in prediction. And there is a heck to be predicted in social interaction. It's just like we know that it's very important in predicting and generating models related to motion. We think that it's basically going to do the same thing across a number of different domains. And so that's definitely one of the areas that we're going to go next. There are 3 studies that I wanted to cover here, and since I'm going to, I think it's late, I'm going to just tell you what the studies are without going over first in the outline. But this next study is actually focused on connectivity of the salience network. The salience network is considered one of those higher level network that is thought to be very important for allocating attention to what is most relevant in your environment, whether that is internal stimuli or whether that is external. This is the network that it's supposed to basically either make you focus on what's out there or on your own, body proprioception and whatnot. And the good thing about this network is that we know from prior work that it actually is an early developing network, and then we can see it very early in development. And so, based also on some studies that we have done in older individuals with autism that have shown that salience network connectivity predicted, if you will, the extent to which an individual with autism show sensory over-responsivity, we thought that focusing on this network might have been able to tap into some of these issues way before babies are able to give any overt manifestation of the fact that some stimuli in the environment are particularly aversive to them. Why were we interested in looking at sensory processing atypicalities? Because we happen to believe, like many others do, that while autism is characterized ultimately by or defined by social deficit, that those may actually be the results of earlier sensorimotor atypicalities that can eventually cascade into an atypical pattern of social development, and how the mechanism by how this happens could be that there is decreased attention to or social attention because there are other systems that are stimuli that are driving attention away from what should be the most important things. I'm sure that I don't need to speak to this group about the disruption in those early attentional biases that are typically present in typically developing babies, but that seems to be disrupted in autism. So this was work that was done by another graduate student in the lab, and we basically focused on this initial study and the scan at 6 weeks. We had eye tracking, as I mentioned earlier, all the way from 3, 6, 9 months of age. It was done both on both the Sesame Street cartoons and another video. We chose this because there was already a study in neurotypical development done by Scott Johnson. So trying to catch up here, we got some beautiful maps of salience network connectivity between across the two groups. Again, at that lower threshold would show more of that frontal region that is a signature of the salience network. When we then compared the groups, we did find that the high likelihood groups actually show greater connectivity between the hub of the salience network, which was our seed for the analysis, and that is the anterior insulis, and a bunch of other areas involved in sensory processing, like somatosensory and also auditory cortices and the thalamus. When doing the opposite comparison, we showed that the low likelihood or typical likelihood group had greater connectivity between this hub of the salience network and frontal regions that are actually higher level region implicated in social processing. What was most exciting to us is that we decided to see whether there was a relationship between these two different patterns. So for all of the babies in all of the groups, we extracted a parameter estimate that indexed the strength of the connectivity between their anterior insula, the hub of the salience network, and those somatosensory region that show greater connectivity in the high likelihood group, and the strength of the connectivity for those regions where the low likelihood group had greater connectivity. And then we ran a correlation. We did show that there was actually a complete trade-off. That is, the babies that had the highest connectivity with the somatosensory or other sensory cortices had the least connectivity with this frontal region and vice versa. So this, to us, was exciting because we thought that it was finally some empirical evidence showing that there is a relationship between attention to social relevant stimuli, if you will, versus more processing of a non-social stimuli. So we obviously had the data that I talked to you about, so we explored that hypothesis a little bit further, and you can see here that in comparing the typically developing infants and the other one, there was quite a bit of variability. There were actually no significant differences in the amount of time that they spent looking at faces. However, there were differences in what we see when we entered their the behavioral measure was the slope of change. So we know that during the first year of life, babies pay more attention as they get older to faces, and that predicted only in the typically developing infants. Okay, so it's kind of weird what you are predicting. So because it was the brain at 6 weeks that predicted the extent to which they showed changes in look-in time to faces. So the babies that had the steepest increase, that got to look more at faces from 3 to 12 months, were the ones that had the stronger connectivity between the hub of the salience network and a similar region to the one we've seen in the between group comparison. Then we also looked at the relationship between the salience network connectivity at 6 weeks with the initiation of joint attention, and we found again only in the typical likelihood group that there was again a prediction. This time it was still that prefrontal region, but also some of the ventral striatum that showed that the one that had this increased connectivity with this region were initiating joint attention at a higher rate at 12 months. And then when we also found some brain behavior relationship in the high likelihood group that actually showed that increased connectivity between the salience network hub and primary and secondary sensory cortices. In this case, it was auditory cortex, was related to parental reports of sensory sensitivity between 6 and 12 months. And lastly, we also saw that increased connectivity between the insula and the basal ganglia and amygdala and thalamus were also predictive of worst outcome at 12 months of age. I mean, increased symptomatology, I should say, at 12 months of age. And these are influential outliers, actually kids that ended up getting a diagnosis, but even if you remove them, there is still a slope in that direction. Okay, so I think I mentioned that there was some connectivity with the thalamus. The thalamus is obviously an important critical sensory gate, right? So it made sense if we want for us to look at thalamocortical connectivity next to really see whether that is another area where we can detect early differences. And this is Arti Nair, that was a postdoc in my lab at that time, and she had done a lot of work on thalamocortical connectivity in older youth with autism, and so she was happy to jump in. The approach she took was actually to start from the cortex, so all of those colors maps, you're looking at different cortical region, and then see how how these regions were connected to different regions of the thalamus. And here is what she found, and the between group comparison showed that there was actually decrease connectivity in the high likelihood group, so the typical likelihood groups in the prefrontal cortex, and increased connectivity, stronger connectivity with both occipital and motor regions. What we also did, we did kind of like a reverse pattern. Once we got this difference, we took these areas where we saw the difference, in particular in the prefrontal cortex, which was the most robust finding, and we saw where is that area connected, because our prefrontal lobe was fairly large, right? It was the entire prefrontal cortex, so we were interested in figuring out if we could zoom in, and indeed what we found that this reverse thalamoprefrontal connectivity mapped onto the same kind of right inferior frontal gyrus frontal region that we have seen being implicated in the other studies. And importantly, what we saw is that the extent of this thalamic prefrontal connectivity at 6 weeks of age actually predicted time spent looking at faces at 6, 9, and 12 months of age. And what you see here is that it was the case for the typical likelihood group, but not for the high likelihood group. We have then continued to look at thalamocortical connectivity, in part because this is an area where we have both relevant expertise and interest in characterizing, because in other work done by Shula Green, who was a postdoc with me, and now she's junior faculty in our department, we call her the sensory over-responsivity queen, because she pioneered the study of this type of symptoms before, I guess she really started, when there was very little attention being paid to this. And this is some work that was done by Lauren Wagner, who is one of my current graduate students, and she now took off where Arti, okay, I should not try to use idioms when English is not my first language, but she basically picked up right where Arti left off, because Arti focused on the 6-week-old babies, but she instead looked at the connectivity in 9-month-old infants, and the approach that she took was different. In this case, we started with this thalamus as our seed, and this is just the overall connectivity maps, and they look fairly similar in between the two groups, but there were actually some differences. And what you see is that the high likelihood group had stronger, had underconnectivity, that's why it's in blue, with some of the prefrontal regions. So that is kind of like confirming, in a way, using a different methodological approach, and 8 months down the road, that you still have this pattern of underconnectivity between thalamic regions and prefrontal cortex in babies that are an increased likelihood for autism. And what we then did in this group, because we had measures of their sensory processing atypicalities, we use that measure in a bottom-up analysis, where we're basically saying, okay, this is the variability in our sample. Do we find regions in the brain that varies according to the parental reports of sensory over-responsivity? And indeed, we did find that higher SOR for sensory over-responsivity was correlated with stronger activation. Guess where? Again, primary sensory regions like auditory regions, and the thalamus, and somatosensory, as well as the thalamus. And the less symptoms of sensory processing atypicalities the parent reported, the stronger the weaker the connectivity was with the some of the higher level region, like the default mode network, which is another one of those intrinsic connectivity network. So again, different populations, slightly older, similar patterns of disruptions. We follow the same approach we did in the salience network, where we extracted parameter estimate for all the babies from connectivity from the thalamus to the region where the high likelihood babies showed less connectivity, or rather the high likelihood babies show greater connectivity, and vice versa, the region where they show the least connectivity, and the two were again clearly related to one another. So it really seems that you either have a brain that is kind of wired to pay more attention to what is out there in your environment, or you have a system that it's really more focused on your own sensory perceptions. So, in conclusion, we do clearly see individual differences in both brain structure and brain function that can provide what we hope some reliable marking of autism in the first year of life, but clearly no one of these measures is at the point where we can use to predict. But my hope is that together with both behavioral measure and as well as genomic data, combining all of these sources of data can ultimately allow us to identify babies that will show atypical developmental trajectories very early on, and obviously well before the behaviors, the symptoms emerge at the behavioral level. I like to think that even if we are not able to really contribute to this one biomarker or a set of biomarkers that can really do the job and be deployed out there, that at least some of this work has helped us think about the mechanism, and that may actually early in development send some infants down a less than optimal trajectory. Why I think it's important is because the brain is so more plastic early in development, in the first few months of life, that I like to believe that it would take much less to tweak the environment to, again, steer development down a more optimal trajectories. And with that, I have to acknowledge not just my collaborators, the funding agency, and this large group of trainees and research assistants that have spent so many nights trying to get this data from the babies, and of course all of the families that are participating in our studies. And I'm gonna thank you for your attention and leave you with some cute pictures of some of the babies that took part in our studies, and I'm happy to take any question. Thank you.
My name is Sumi Shankar. I'm one of the clinical geneticists here, but my question is not going to be about genetics. I was wondering, how did you get these cute babies in the MRI mission?
Okay, I have actually deleted a slide that showed some of what it looks like, because I thought at least some of you here might have had some experience. It's actually not that hard at all when they are 6 weeks old, because they do sleep a lot, and all of this is done during natural sleep. At 9 months of age, it's getting a little bit trickier, because by then they typically do have more established, day-night cycles, but they are also very aware of their environment. So when you take them, even if it's their nighttime, and then they are in a very weird location and an environment, they may struggle with going to sleep. But we have how we do it. We have incredibly motivated parents, and we have incredibly motivated research assistants and trainees who are willing to put in an enormous amount of time. We tell them, try to make sure that your baby doesn't fall asleep in the car, because if they just had a nap, that is bad news, but sometimes it cannot be prevented. And so we have an arrangement where we always scan at night where there is nobody else doing scheduled to scan after us. So we're basically willing to wait as long as we need in order for them to fall asleep. Some sequences tend to wake them up a little bit more than others. Sometimes there is a break where the parents work hard to put the baby back to sleep. Sometimes it's a no-go and we bring them back for another visit. We do everything we can to protect their ears. They wear silicone plugs, and they have the mini-muff that are also used. Yeah, I have a slide here. Let's see. You see, like, so there is some plugs underneath, and they have this, and then parents, we have rocking chairs, we have changing table. You know, we thought about buying one of those swingers too. We ultimately never did. But I want to tell you that when we started this, and I was actually the person talking to the families to convince them to participate, at some point I got convinced that we were never going to be able to scan the very babies that are going to be more likely to develop autism, because some of the parents would tell me, okay, so I tell them we're going to put these things in their ears. They go like, oh no, you cannot do that. My baby hates to be touched, et cetera. And or we would tell, there was one family that said, okay, do you have a swing? And I said, well, no, but you know what? We can get a swing if that's what helps your baby fall asleep. Like we have a rocking chair, et cetera. And she goes like, no, but can you put the swing in the MRI? And I said, no, I don't think we can do that. And she said, because my baby only sleeps in the swing, literally. And I said, oh my God, this is peculiar, and we actually know the sleep issues is one of the things that really is associated with autism. And so I thought we may not be able to put in the scanner successfully precisely the babies that may have a higher likelihood of getting a diagnosis later on. But parents of infant SIBs are very motivated, and they really are very generous with their time. So.
I have had families ask me if I can build a hammock inside the bore of the magnet, which we have not yet done. But so this was really, really interesting, and I know the amount of work that has gone into this, so I really appreciate it. I'm just wondering if you can speculate at all. So, I like how you combined everybody into one group, and you're looking for early brain markers for later behavior. Can you speculate at all about differences between the high likelihood that don't go on to have autism? And I know some of them will go on to have other developmental delays. And so I, in some ways, I do really like putting them all together. But
yeah,
I'm also wondering,
yeah,
what your thoughts are. And then I have a part. Well, go ahead.
No. Okay. I just want to make sure that I don't forget, but I want to hear your second part. So I like to think that there is some genetic risk may confer some liability early on, so that maybe some connection are not happening the way they should. But I mentioned plasticity, and I put the word resilience also in there, because I do believe that coming in with maybe similar risk factors, that the environment may be, the interaction with the environment may actually scaffold or support a more typical development, and that's why some babies, even having the same maybe genetic risk, are able to develop more typically. Because if that was the case, then there is really hope, right, for intervention later on. It is also the case, maybe that, no, it's definitely the case, we do put in this group, there is the heterogeneity, that it's due to maybe you're being able to overcome some hit, but there's also some of the babies that do not get a diagnosis, or that do not present with any symptom whatsoever, that have a very clear typical development. They may just not have really the same level of genetic risk. They may have a sibling who maybe have developed autism because of some other idiopathic, reason, maybe, being born very prematurely or whatever. And so that's part of it, maybe.
It makes me think of some of your other work where you've looked at polygenic risk scores and brain imaging, and do you have saliva in? Can you get
we
DNA
have
in
been
these
collecting that, and again, at the end of the first 5 years, it was definitely too little a sample to do much about it. As we ended now the second cycle, we are in a better position, except that, of course, our recruitment took a hit because of the pandemic. But I was recently talking to Dan Geshrin, and he told me, so how many do you have? And I said, we have 100 and whatnot. And he goes like, we can do it. And so it's work in progress. We're definitely going to be looking at that. And actually, one of the aims in our grant was to compute several polygenic risk scores, like for sensory over-responsivity, and not just using the autism, but basically all of the polygenic risk scores that you can think of related to symptomatology that is present in autism. So stay tuned.
You'll have to come back to tell us about that. And then just really quickly, I'm sorry, extraaxial fluid, have you ever looked for it in your babies?
We haven't. And so it would be very interesting to look at also, because I think that there is some work showing that that may relate to sleep issues. So yes.
Yeah.
I have to say that because I started my neuroimaging career more as being as a functional person rather than a structural, and we have typically put our MP range, our structural last. And so we have, sometimes the baby would wake up before we were able to collect it.
But I prioritize the other way.
Yeah, yeah.
But yeah, I mean, you might have enough to look.
I think so.
So,
You're right, yeah. I just need more students.
I am. Well, thank you very much. That was really amazing. And it's a labor of love, I can tell.
Thank you for this incredible work. It's inspiring to see the amount of effort that, and that the story is incredible in terms of things that you can measure during sleep in an infant, predicting the later behavior. So I have two questions. One is, do you find that the anatomy that you detect as an early infant is present and correlates with the behavior later on? Or are you, so you're basically identifying brains that are going to grow in a particular way and stay that way, if you have those data? That's question one. Question two, we're all in academia so verbal. So you're talking about statistical learning, about auditory patterns. There are auditory patterns all over that are helping us build a world that integrates what we see with what we hear about actions in the world. And you would think that since I love, we've been long proponents of this basic sensory processing giving rise to ultimately social consequences. Do you have ideas about the world building that the infants are doing in terms of the actions that are going on around them and their praxis for understanding how to interact with the world separate from or along with language development?
Okay, I couldn't quite understand. So do you have an idea, the last part where you said, do you have an idea how the babies are creating, I'm sorry, what?
They're world building.
Okay.
In other words, if we all are figuring out what the planet is.
Right. I don't know that I'm going to give you the best answer to this very, it's a big question, but two things. At 6 months of age, they don't have an enormous behavioral repertoire, right? But that's not to say that they're not active constructors of their the environment in which they.
Yeah, I mean, I'm thinking like, what Esther Thalen used to talk about, the pre-linguistic propositional calculus of,
right.
Trying to overcome reflexes and understand there's gravity, whatever they're.
Yeah, no, I don't know. I wish I could tell you a little bit more about, but when it comes to motor behavior, we're collaborating with Rujita Bath at UCLA, who has been using sensors that are put on the baby's limbs to actually quantify their movement, very early in our babies. And we have some interesting, she has developed a measure which is called motor complexity. And it basically, we're just now beginning to explore the relationship between those motor measures and both behavioral indices of symptomatology, but more particularly with the brain. So that would be very interesting. The other thing that I think we should be looking at, and I'm using the royal we, not necessarily us, but I know that some of you here are doing it, although Sally Ozanoff has already left, is to really take another look at mother-child interaction, what's happening, not going in with our schemes as to what is the important thing to look at, but rather use artificial intelligence, right? And use sophisticated machine learning algorithms to actually tell us what is different. And I know that there are studies that have found, even with our more canonical coding system, differences also in the way parents may interact with these infants, which is obviously very important because it's in a dyad, you respond to what the other person is doing. So you may not engage in the same way if you have a child that is less engaged, engaging less with you. And the thing is that my theory of how this whole thing fits together is really this, that something in the wiring of the brain, maybe because of the genes, maybe because of any insult, perinatal or antenatal, it doesn't matter, is a lot of the genes are implicating, right, in measures that, in things that will impact the cortical architecture. So you have something that maybe wires slightly different, and then that may cause a disruption in this attentional biases, which really are paramount importance. And it's very, very hard to quantify, to know how a baby perceives their environment before they can actually do things like, covering their ears and or closing their eyes, or before they can start pulling their clothes off. So I think that there's progress in this arena. There are some folks that are trying to, they are developing more sophisticated ways of assessing responsivity to stimuli without only relying on the parents' report. And maybe there are other ways in which we can tap into that. But I don't know that I answer your question ultimately.
It's fine. It's in the domain. And
Thank you. I'm Andrea Schneider. I'm more on the clinical side, working with social skills groups and parents, et cetera. And one thing that we mostly focus on is communication skills, like the back and forth, et cetera. And on and on and on, it comes up that this naturalistic conversation of how is a pragmatic back and forth in a conversation is nearly impossible for our kids to achieve. So when I then talk to the parents is, might that be something that is linked to the implicit learning, that you have to make it very explicit for them to get it. And now seeing your data is like, yes, we have an indicator for that super early thing. But now thinking, would it make sense to do a super early intervention for this naturalistic back and forth? Yeah. And how could that look like at that age? Do you have an idea?
Yeah, no, I totally believe you. I would say that when kids are older, the best we can do is, intervene with more explicit concept and training. But I think that if we were to move intervention very, very early in development, it would take less effort. It would be different, and more likely you can re-engage the systems that are supposed to subserve those processes in an automatic way, right? For us, we don't go about looking at a person's facial expression and start saying, okay, well, their eyebrows were raised, the corner of their mouth were going in this direction, et cetera, whereas they obviously do that. I do like to think there's obviously the field is moving in the direction of doing this. I don't want to call it preemptive intervention, but they're really, starting to do a lot of work in this area. Again, I'm not a clinician, and far from me to want it to go anywhere. The notion that the parents' behavior is it's maybe a causative. May play a causative role, but I think that if parents are aware early on that maybe their baby has difficulty predicting their environment, or they are particularly sensitive to some environmental stimuli, or it's harder for them to focus on certain aspect in their environment, that even if it's natural to maybe be less responsive to somebody who maybe doesn't smile back, or that it's not doing so much of this back and forth, I think parents could be taught or have, trained on how to increase the frequency, to make things more predictable, to take away some of the competing sources of attention, the distraction that is there in the environment, and to kind of really help their baby hone into what is most relevant, which is what other people, other human beings are doing or saying, right? But yeah, how do we go exactly about doing that? That's a different story, but I'm optimistic actually about that.
My name is Kim Walters. I'm just from the community and come and enjoy these talks and learning about the updated research. I did have a question that has been going on in my mind and just through observation of my close friends and family members, that in dual language homes, when you have one parent who's speaking fluent Spanish, another parent who's speaking fluent English with the young child, it seems to be a pattern of delayed language. And I know that you were doing some research on delays and how it could possibly relate back to autism. And I didn't know if you saw any correlations with dual language homes and being raised in that environment, any delays or have any correlation with that.
Good question. I don't know that we have a large enough group of babies where we can tackle that in our sample, to be honest with you. We do have in LA quite a range of exposure, like a lot of our babies are sometimes exposed, to multiple languages in the home. We have, for some of the studies, like the word segmentation study, we excluded them from the analysis because we didn't want to have that additional confounds, if you will, because you're right, there is some empirical evidence that shows that for some babies exposed to multiple languages that there is a slight delay in the rate in which they start talking, but ultimately they catch up in it. And there is actually a person at UCLA, Lucina Uding, which is quite interesting, looking at bilingualism in youth with autism, because some folks have argued that the bilingual brain has exercised this ability to switch, and basically that their executive function may actually be better because of this experience, switching between codes. So maybe that's something that we could look into that. I have to see what the numbers are, and I expect that with one of the large-scale study that I alluded to, there will be we will be well positioned to looking at the neural differences in the neural substrates of language learning in babies that are growing up in a bilingual environment. But with regards to autism, I think that it's hard enough to find enough babies that are in increased risk that to also achieve a large enough sample to look at any difference between the one who grow up in a monolingual environment and the ones that are not. It's a bit challenging, but we'll see how many we have.
I agree. That would be amazing, an amazing study to do. Previously, I was a special ed teacher, and I had many students that came from bilingual homes. And, it was funny because they were still really sneaky. If I wasn't in the room, I would hide, and they would tell each other answers in Spanish because the rest of the staff didn't know Spanish. And they're like, oh, good, Miss O isn't here. And then I'd pop up, they're like, oh, hi, I was just kidding. And so, eventually I would see them catching up, maybe they were, they were catching up in their sneakiness and knowing when to code switch. And
Right.
I noticed at least in my classroom and with the kids that I worked with, they weren't as easily able to realize when they needed a code switch. With the other bilingual folks, so they would continue speaking Spanish with me, and they didn't understand why no one else understood. I'm like, you're speaking in code, and they're like, oh.
Yeah.
Oh, so
Yeah.
It was that part I at least noticed. I'm like, that's where they kind of got stuck, but that
Yeah.
would be so cool if, that study got up and running, because you see these things and you wonder what more is there to it.
Right. No, I agree. Thanks.
The UC Davis Mind Institute was founded in 1998 with the promise to reduce and prevent the disabilities that can be associated with autism and other neurodevelopmental conditions. Every day, our clinicians and researchers make progress on that promise. Our groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other conditions associated with disability are helping affected individuals achieve their fullest potential. Please visit our website or our social media platforms to find out more about current studies, upcoming events, and how you can help make a difference.